Hi guys, on to chapter 12, objective 6, talking about Korsakoff syndrome and Alzheimer's disease. Well, let's start with Korsakoff syndrome. Korsakoff syndrome, it results from a long-term thiamine deficiency. Thiamine is a B vitamin, and it comes from, I think we've talked about it before, people who are alcoholics who don't eat don't tend to get enough vitamin B, and then that results in this deficiency. And again, it has to happen over a long term of time, a long period of time. They get damage to the dorsomedial thalamus, which would normally provide input into the frontal cortex. So we start to see some frontal cortex damage. From that, you start to see anterograde and retrograde amnesia, problems remembering things from after the damage, and problems remembering things from before the damage. A lot of times people with Korsakoff's disease will tend to deny that they have any problems with memory, and what they actually do is they make up stuff to fill in the memory gaps. They just kind of make up stories when they can't remember things, and that's called confabulation, and that's particular to Korsakoff syndrome. Another problem with memory is Alzheimer's disease. Alzheimer's disease tends not to afflict young people so much, but as you get older, it gets to be a lot more common. So age 85 and up, about 50% of the population has Alzheimer's disease. So the longer you live, the more likely you are to get it. We do see both anterograde and retrograde amnesia in Alzheimer's disease. We get short and long-term memory loss. We get confusion, depression, psychosis. It tends to be progressive, meaning it gets worse and worse over time. There's no real treatment for it. We see better procedural than declarative memory, similar to HM. You can learn how to do things, but not remember events. The memory tends to wax and wane, and as you might remember, that the damage is in the basal forebrain, which is kind of an area that sort of wakes up the brain and also puts the brain to sleep. And so with greater arousal, the memory tends to be better. The memory problems, or the main source of the brain damage in people with Alzheimer's disease, is a loss of acetylcholine caused by degeneration in the basal forebrain there. And it's caused by there are a couple of different proteins one is called amyloid, and that tends to build up plaques around the neurons, and then tau, which tends to build up these tangles inside the neurons, and both of them cause neurons to have problems firing and eventually cell death. And again, those are mainly on the acetylcholine neurons in the basal forebrain. There's not much in terms of treatment for Alzheimer's disease. Just like all brain diseases, we can't really do much about the cell death, but there are drugs that will increase acetylcholine if you still have the cells that will produce it. And so, but unfortunately, it's not a cure. The disease continues to progress. We do see different dietary things that might in fact, improve Alzheimer's disease. Turmeric tends to limit amyloid production. People get amyloid, by the way, all throughout their lives. And some people, prior to getting Alzheimer's disease or being diagnosed, those that are more likely to get it will have greater amounts of amyloid. So anyway, some people will eat turmeric as a supplement because of that, limiting that amyloid production. Okay, last thing we're going to talk about is the mechanisms of long-term potentiation, or LTP. So long-term potentiation is really where the engram is in the brain. It's what's happening when you learn between the cells. And so remember, when you learn something, it's not like you're getting any new neurons. There aren't any new neurons being formed. What's going to happen when you learn is that you're going to get changes in the synapses between cells. They're going to act differently. And there you can get a strengthening or a weakening of synapses. Well, we look at learning primarily in an animal called the sea slug, or a plesia. And we look at it there because they have a few big fat neurons that you can see really well. Okay, so let's see what happens in the plesia. there's a couple of different kind of learning that we can figure out pretty easily. One is habituation. If you touch the slug, it withdraws. But if you keep touching it, it stops flinching like that. It recognizes that, you know, it doesn't matter if you touch it, you're not hurting it or whatever. What happens there is that the sensory neuron that senses the touch is still continuing to respond. It's still sending information, but it doesn't excite the motor neuron. And so what happens is that there becomes a weakened connection between the sensory and motor neuron. At first, the sensory neuron excites the motor neuron and the animal flinches. But after doing it over and over and over again, all of a sudden this synapse becomes weakened and the motor neuron does not work anymore. So that's the weakening of a synapse that involves learning. The animal is learning that it's not dangerous. Okay, then there's another kind of learning that we can see in the sea slug. It's called sensitization. And that's after a very strong stimulus, like an electric shock, the slug will become very sensitive even if you touch it. And so the shock, as it turns out, will cause an interneuron to release serotonin at the sensory neuron synapses. And that what happens is that causes a sort of prolonged action potential, more neurotransmitters released to the motor neuron, and then we get a stronger response of the motor neuron. So what's happening there is the synapse is becoming more sensitive. So, you know, this electric shock causes this motor neuron to react wildly. Now the touch comes along, and this synapse is so sensitive, the motor neuron also reacts wildly. So this is just a little diagram. If you want to pause, you can kind of go through what's going on there with sensitization. Okay, well, I want to talk now about long-term potentiation in mammals, which is something that it probably accounts for most of the learning that we experience. It's the strengthening of synaptic connections. Sometimes we say cells that fire together wire together. Long-term potentiation is when bursts of an action potential leave this neuron highly responsive to similar situations. So this synapse becomes strengthened. If we use it over and over and over again, it becomes very responsive, very strong. Okay. In the hippocampus, where it's been studied mostly, LTP, or long-term potentiation, is going to depend on glutamate. So to understand what's happening here, you have to understand that there are two types of glutamate receptors. One is the NMDA receptor, another is the AMPA receptor. The NMDA receptor is really hard to activate. It's not as sensitive. The AMPA receptor is very sensitive, okay? Now, the reason why the NMDA receptor is not sensitive is because it is blocked by magnesium. And the only way to get that magnesium out of there is to keep working at the AMPA receptors until the cell hyperpolarizes enough to knock out magnesium. And then that will allow calcium to enter, and then we're going to get some changes based on that calcium. Okay, let's look at this. What's happening here is that this axon is releasing glutamate repeatedly over and over and over and over again. We're getting these action potentials. Here's your AMPA receptor. It is activated very easily, okay? So it gets stimulated over and over and over again. But the NMDA receptor has magnesium in it. And it's not until enough of these AMPAs go off, the dendrite gets really depolarized, that then this magnesium pops out, and now we get the activation of the NMDA receptor. So in order for long-term potentiation to happen, we have to get both of these activated, first the AMPA and then the NMDA. Now, when that happens, we start to get calcium entering into the postsynaptic neuron. So let's take a look at what that calcium does. Calcium activates proteins that will increase the sensitivity of the AMPA receptors. So now they're very sensitive, even more sensitive than before. It will also cause production of new receptors and will cause growth in the dendritic branches. So now you have more places for receptors. So what's happening here again is that all of the activity in this neuron has now strengthened the synapse here. So it's very sensitive, very sensitive. So when you get a little bit of activity in one neuron, all of a sudden the next neuron is going to go off. Okay, so what this is when basically when you learn things and you're rehearsing it over and over and over again, what you're doing is you're releasing that glutamate repeatedly, repeatedly, repeatedly. When you're studying for this class, you're releasing the glutamate repeatedly, repeatedly, repeatedly. And this connection here, the synapse is slow to begin with. It's just AMPA. But eventually, if you do it over and over and over again, if you practice something over and over and over again, you're going to get a stronger connection here in the synapse. It's going to become more sensitive. You knock out that magnesium and the NMDA, and all of a sudden, now you have learned it. You have committed it to long-term memory. And this is a long-term change in your synapse. Okay. Well, there are a few other things that happen in these synapses, and those are presynaptic changes. So not just the postsynaptic neuron here, but also the presynaptic neuron makes some changes. So let's take a look at those. Lots of stimulation will cause that postsynaptic neuron to send back a retrograde neurotransmitter. So normally neurotransmitters go in the direction of axon to dendrite, but in this case, it's going to go from dendrite to axon. It's a retrograde neurotransmitter. It's going to lower the threshold of the presynaptic neuron. So now that presynaptic neuron fires with less stimulation, it's going to expand the area of neurotransmitter release, and it makes it release more neurotransmitter. So all of this is, again, strengthening that synapse. So as you learn physiological psychology or history or abnormal psychology or whatever you're studying right now, what's going to happen is you're going to have to practice, 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 practice. And at first, these connections are going to be weak. They won't respond very well. But eventually, that connection will become intensely strengthened so that when you think about something, automatically the answer comes to you. And that is really the essence of learning in the hippocampus. It's the essence of long-term memory in the brain as far as we know it. I hope you guys enjoyed the presentation, and we are on next to our last chapter, getting close to the end.